Hi, everybody. I had a feeling I want to share with you some of the background that will explain better why we do things in lab the way we do it. And this, I would like to share my screen and show you the outline of my quick presentation. Just I'll tell you why we make yogurt in a lab and what's in it. Then I'll talk about what microbes do in human and how we keep them in check. Then how do early childhood and diet affect our microbiome? Finally, food preservatives and the history, very brief, of FDA establishment, and how can we test food preservatives in the lab? Yogis. The list of the microbes in yogis are listed right here. The top 2 are essential for making yogurt. The rest are added as bifidobacteria, helpful and beneficial bacteria that also called probiotics. The last one, Lactobacillus Rhamnosus, is my favorite, and I'm going to explain you why. And by the way, all of those are found in the healthy human gut. Something about Lactobacillus rhamnosus is very exciting. This bacteria have been shown repeatedly in several animal models that it regulates emotional behavior. In mice and rats and in chickens. Human studies have been inconsistent. We are complicated human beings, and our emotional health is even more complicated. So it's no surprise that a single strain of bacteria cannot fix all of our problems. But in simpler systems, it's been shown that this bacteria interacts with the vagus nerve and enhances release of GABA in the brain, reducing anxiety behavior. You will be very excited to find out that this is a bacteria that we streaked on the plates last week to test in the disk diffusion assay. Now, some of you mentioned that microbiome in human is important for establishment of immune system. Yes, indeed, in fact, this is how it works. I took this picture from this article from 2019. It depicts a layer of mucus that covers our gut. And mucus is a universal system to protect sensitive membranes of animals that have to interact with environment by either exchanging gas, exchanging nutrients, or releasing waste. So animals from anemone to human use mucus. And mucus is a fantastic substance that is holding in itself 10-fold amount of viruses, or more accurately, bacteria viruses, which are called bacterial phages. In fact, almost 6% of non-human DNA are those bacteriophages in the mucus. What do they do there? Well, they fight bacteria. And in the process of this fight, they interact with our immune system. In this image, you could see that our immune cells stick out the receptors into the mucus and sense the product of breakdown of those phages. And based on those, they build antibodies and prepare to fight any infections that may occur. And these processes start taking place from birth, as soon as we interact with our first microbes. So that brings me to the picture on the right side. Which bacteria do we interact with when we get born? Well, in the natural way, how it used to be in most of the animals, babies come out from the birth canal, where they pick up certain vaginal bacteria. And this is the first bacteria our immune system it will interact and learn how important it is. Babies that are born through cesarean section, they will interact more with bacteria that is more common to the skin here. And they will learn in a slightly different manner. The next step is what the babies eat. The babies that are fed on breast milk will support certain microbes in their gut. In fact, human milk has up to 200 different HMOs, oligopolysaccharides, that are indigestible to babies and specifically made to feed those beneficial microbes. Formula doesn't have those. So the different kind of microbiome will grow. And those things getting recognized just recently. This paper came out in 2020 of importance of breast milk in the early development of immune system. But that's not it. Another important factor that regulates our microbiome inside is the diet. And as you can see, American diet is not great. Americans eat a lot of animal food and a lot of processed food. Why is it bad news? Because fiber is extremely important in the gut. It supports fiber degrading microbes, and those microbes tend to maintain a thick mucus layer in the gut. Once we don't have enough fiber, the mucus layer will wear off, and that may lead to certain inflammatory disease in human. That's why culturally, countries that contain more plant food has usually less inflammation based chronic disease. But besides fiber, processing food almost always involves addition of certain preservatives that help this food to stay fresh longer. And this is a great thing if you want to prevent food poisoning, but not so great if you want to take care of your microbes. Those are an example of wine, jam, and ketchup. All of them contain preservatives that we're going to study in the lab. And those preservatives date back more than 100 years ago, when Dr. Harvey Willey, a chemistry, he was very concerned about what Americans eat. And he set up an experiment called Poison Squad, made up of about a dozen healthy men who started eating at the common table. And at every meal, they would eat certain type of preservative with it. And during the experiment, they developed certain health conditions. After this highly famous experiment in 1903, a number of chemicals were banned from food, including salicylic acid, formaldehyde, and borax. Benzoate and sulfides passed. And that was the very, very beginning of FDA in the United States. Not so fun fact about FDA. Around the same time, Coca-Cola was also forced to stop adding cocaine in its drinks and substituted it with a very high amount of caffeine. And in 1909, same chemist, Wiley, he took Coca-Cola to court for selling drinks that was still highly addictive, especially for kids, and he lost. And he was forced to leave FDA afterwards. And as you can see, Coca-Cola still sells drinks that we consume. In lab, we will be testing 4 different preservatives, which are generally recognized as safe, meaning that experts consider them safe if used as intended. It's sodium benzoate, sorbate, propionate, and sulfite. And instead of healthy volunteers, we're using disk diffusion assay and yogurt fermentation methods. This diffusion method, we're testing 3 different chemicals on each plate with 4 different bacterial strains. What are those 3 chemicals? The first one is a first aid antiseptic vaccine. It's designed to kill bacteria more or less evenly, and we use it as a positive control. Lactic acid is a compound produced by lactic acid bacteria, and it aims in food preservation by fermentation. And the last chemical you'll add is your assigned preservative, and you will create 3 replicates of it on the plate. The 4 different bacterial strains we're testing will be E. coli, a common food contaminant. We're using non-pathogenic strain. The second one will be our favorite now, Bacillus Rhamnosus, and it's used as an example of a good gut bacteria. And the last two will be just the usual bacteria commonly found on produce. The question you will try to answer is, will different bacteria react differently to various antimicrobial agents? If so, why? Try to figure out this week. The second method we will be doing is fermentation method. It is used to model system to mimic the microbial interactions with a preservative in the human gut. You set up 4 different jars. One jar is milk and yogurt. It is used as a positive control. Second one is just milk. Random bacteria will grow and spoil milk. The last 2 jars are 2 replicates with milk, yogurt starter, and a preservative that been assigned to you in the concentration approved by FDA. You will analyze the data this week and we'll try to figure out, will the milk in experimental jars get preserved, spoiled, or fermented? Try to figure it out this week and explain what happened. I'm looking forward to your presentations next week.